Blagodaria, which, according to my Bulgarian friends, means good morning. At least I hope it means that. I'm an elderly microbiologist, and there are a number of problems in microbiology which interest me. The question is, what does one do about them? So let's have a look. Okay, so this is just a little montage. One of them is a cartoon which I'm sure you're familiar with, which is the man underneath the lamppost explaining to the policeman that he's looking for his keys, and the policeman asks him, "Is that where you dropped them?" And he says, "No, but that's where the light is. That's where I can see." And this is a perfectly respectable way to do science. You look in the area where you can do things. Of course, the problem is, for a biologist like me, what if you suspect that the keys have fallen into the water? And if you can't swim, then you find yourself in the position of many biologists like me. What do you do about it? Do you pretend that the solution cannot be there? Or do you hope that the policeman is actually a friendly physicist working on water who's going to guide you through the subject? Okay. But first, of course, I say, what is the question? That's always important. What is a question that biologists should be answering? Well, here on the left, you've got another, oh, sorry about that. Here on the left, you've got another sketch by David Goodsell, in which this is supposedly a section of E. coli here, with the strands in red are DNA, the squiggly white bits are RNA, and some of this RNA is being translated by ribosomes to make proteins. The ribosomes are these sort of red turkeys. Okay. And as you can see, this is a pretty crowded environment. Surprisingly, there's no water present, and actually it's relatively unstructured. That's what I observe. Now, going on in here, you've got a large number of constituents, actually a huge number of different constituents. Many years ago, Stuart Kauffman, in a wonderful little book called At Home in the Universe, drew his readers' attention to the fact that if you have, for example, just 4,000 different constituents which can be either present or absent, assuming that this presence or absence is independent, you would have 2 to the 4,000 combinations, or 10 to the 1,200, 10 followed by 1,200 zeros, which is just a huge number of possible phenotypes, and really impossible to get a reproducible phenotype on which natural selection could act. That's a fundamental problem. I've added to Kauffman's original remark, of course, you want to have coherent phenotypes. Coherence is really important in life in general. I'm going to struggle, of course, to be coherent today, but nevertheless. Okay, so I'm going to suggest to you that the answer to Kauffman's question involves new findings and new ideas in biology. I should say new visions, because if I say new visions, you'll realize that many visions that we can have are actually hallucinations. So there's a health warning in what I'm saying. What I'm saying is probably not true, but it's a new view. Okay. So what are the places on one could look for answers? One is in the dynamics of things we call hyperstructures, which are large, spatially extended assemblies of molecules and macromolecules which perform functions in cells, and which we think are the level at which one should look for explanations of the phenotype. And by the way, if there are only 100 or so hyperstructures in cells, the combinatorial problem is reduced to 2 to the 100. So we've got rid of 1,000 orders of magnitude, which is pretty helpful. The second is something called the cell cycle itself. The cell cycle, as I'm sure you all know, is the replication of DNA, the segregation of DNA, and the division of the bacterium, in this case, to make 2 cells. That's a process that can generate, at least I will argue, coherent cells that are different complementary phenotypes. Okay, so let's go on. Of course, the question for this audience is, how might water structures be involved in all of this? And that I'm going to disappoint you right from the start. I'm not really going to tell you. It's for you to tell me. Okay, so cells are made of hyperstructures. Let me try and convince you of that. One way that people study cells, or structures within cells, is to obtain an antibody to an epitope, for example, on a protein, add that antibody to a cell, then add a second antibody to the antibody itself, which is fluorescent, and look to see where that fluorescence is. That way, you can get a distribution of your target protein within the cells. Typically, then, the fluorescent signal fades, and that's the end of the experiment, and if you're lucky, that's your exciting publication. And back in 1988, somebody called Walter Schubert was on holiday on the island of Kos, which is a Greek island in which Hippocrates practiced medicine, rather a long time ago. And Walter, who's a medic, was thinking about diseases and about cells, how to study them, and he was remarking the interplay of the Greek sunlight on the dancing waves in the Aegean Sea. Okay. And it gave him an idea. When he went back to Germany, he did that experiment. Okay. In the slightly weaker German sun, nevertheless the signal faded. And then he did something very strange. He added a second antibody to a second protein, used the same secondary antibody with a fluorescent tag attached to it, and he was able to map the positions of a second protein within the same cell. He again allowed photobleaching to occur, and he added a third antibody to a third protein. And he obtained the distribution of the third protein. Why stop there? So he went on. He plotted the positions of 17 different proteins within the same cell. Okay, and that's what he got. So that's a eukaryotic cell. And those colors correspond to either the co-localization of different proteins or the exclusion of other proteins. So he defined these structures on the basis of the combinatorial profiles of those antibodies, which were all different antibodies to different proteins, which were binding to the same region, and the absence of others. And as you can see from this, there are many different colors corresponding to many different structures. He doesn't call them hyperstructures, he calls them supermolecules or combinatorial protein profiles, molecular profiles. Okay. So we have a highly structured cell, and he can get, by the way, this is on the 2 nanometer resolution scale. He can get a new interpretation of cells and a new way of seeing disease processes. For example, in one of his publications, one can see a structure, kind of a spike, going from one cell, from a cancer cell actually, and going into an immune cell. Okay, so this is called the toponome. It's perfectly respectable, very interesting, and largely unexploited and appreciated. And it's some justification for what I'm telling you about hyperstructures. The question is, for me, can we identify these hyperstructures? Well, let's get started. Well, let me give you an example of an individual one. Okay, cell division in E. coli. The field doesn't call them hyperstructures, by the way. They often use the term somes. So this would be the divisome, the division some. And what you have here is the lipid bilayer of E. coli, various proteins, or at least 10, 15 different proteins involved, and they're responsible for invaginating the membrane, acting together. So a lot of different proteins working together in the same region at the same time, forming a hyperstructure. One of the key proteins in this is the protein FDSZ, which you've been hearing about. Tubulin, this is the bacterial version of tubulin, which forms a very dynamic polymer at the membrane in association with some of these other proteins, and is responsible for providing a force to cause the invagination of the membrane. And here, this is this rather nice experiment where you just have a liposome and FDSZ inside, supplied with GTP, and it's dividing the cell. Okay. So that would be one of many hyperstructures. Okay, so my question for the audience is, could water be involved in the assembly and operation of the divisome? Well, certainly, the composition of the divisome differs from the rest of the cell. Let's have a look at that. Okay, on the left here, we've got a picture of E. coli in which the DNA is in red, so this is a living E. coli, and the cardiolipin, which is a charged anionic phospholipid in the membrane, is present in between the chromosomes. So two identical chromosomes, the cell's getting ready to divide, you've got cardiolipin concentrated in the membrane between them and cardiolipin at the poles. Okay, so cardiolipin helps to define, at least in these bacteria, the site of division from the point of view of the composition. On this side here, we've got, again, we've got E. coli, growing E. coli, we've got DNA, and we've got membrane. And if you look at what's been looked at here is the microviscosity of the membrane, and in this representation here, we've got the DNA from the fluorescent signal of the DAPI, and we've got the microviscosity in the membrane dropping down between the chromosomes where the bacterium is going to divide. That happens at a very, very early stage. Okay, so we've got physical and chemical changes in the membrane associated with division, perhaps triggering division. A question would be interesting would be, in the light of what I've been hearing about EZ, membranes structuring underlying water, would one expect different sorts of structured water adjacent to the regions of the membrane of different composition? And could such structured water play back on the structure of the membrane itself, for example, in a positive feedback loop, and help membranes to invaginate? Okay. Other things one can ask are the reaction rates changed in the divisome? If water is structured differently in the region of the divisome, one might expect, for example, the polymerization of the tubulinite protein FDSZ to be altered. One way perhaps to investigate this would be to see what happens in heavy water, D2O, where the structure of water must be different. And so when I look back at the literature, well, I can go back, I can find evidence that it does change it, so it's consistent with it. Of course, that doesn't necessarily mean that it's due to an effect on the water, but nevertheless, I did find these old papers by Sato showing effects on the polymerization of tubulin, showing effects on cell division. These are, I think, in sea urchin cells, showing effects on the progress of the mitotic spindle, which is being driven by tubulin dynamics. And even an unpublished bit on bacteria. This is on the left, the bacteria, these must be cyanobacteria, being grown in the absence of D2O, and here their morphology is totally different in its presence. That was from Marie-Claire Billis-Funel. Okay, so major effects consistent maybe with D2O having an effect on water, on cell division, and of course a host of other properties, but there could be other explanations. Okay, so now a rather strange question, and fortunately I have an audience in front of me which isn't entirely constituted of cell cycle specialists, because when I ask this to such an audience, you know, what is the function of the cell cycle? Why do cells replicate their DNA? I really have some very strange looks, and people start to take an early coffee break. So, you know, what is the function of the cell? What's a real function of the cell cycle? Well, I have a strange take on this, which is that essentially in life, and this is a problem for all organizations at whatever scale, whether they're human beings, whether they're universities, shops, or bacteria, things have to negotiate between two attractors. One is growing in heaven, and the other is surviving in hell. And the behavior is very different. You have to find coherent solutions. You can't do both simultaneously. You can't, I mean, I work in France, and there's a hereditary enemy is just over the channel. And I tell my students, well, you never know with the UK. They've got their own nuclear deterrent. Who knows whether they're going to drop a bomb on Normandy and Paris and bring the UK into France again, get rid of the French population. You could be pessimistic and think this is pretty likely, particularly in view of recent events. You know, you should spend your life hunkering down in the basement. But if you do that, well, you will survive if the bomb goes off, but you know, you will eventually starve to death. You've got to work. How do you compromise? Okay, so you know, it's like you don't know what the weather's going to be like, so you say, well, I'm going to go for both really cold and really hot weather. I'm going to wear an anorak and shorts. This is not a pretty sight. It's quite incoherent, and you know, you've got to go for one or the other. Don't do both. Okay, so here we are. So heaven versus hell. How do bacteria, which are sometimes I hear things about bacteria which make my blood boil. Bacteria are extraordinarily complex and structured. They made our world. They control us. People ask about the meaning of life, but I can tell you the meaning of life. The meaning of life is that you eat and you defecate in different places. If you do that, you've fed your bacterial population. You've done what you're supposed to do. That's why bacteria invented you. Okay, so bacteria rule, okay? And people say, well, look at the eukaryotic cells. They're so much bigger and more complex. But this is just racism. This is crude racism. The bacterium is like my laptop compared with the ancient computers on which Turing worked. You know, there's no comparison. Okay, so be a little respect, please, for bacteria. They were here long before us, and they'll be here long after us. And every problem we want to solve, every important problem, they've solved it. Okay, so let's look at them. Okay, so how do bacteria solve this problem? Well, as you can see here on this row here, we've got a range of bacteria growing and dividing to produce progeny, daughter cells, which have got very different phenotypes. Oh dear, not how much time for this little one. This one here is a bit strange. This is actually from our lab, and it's taken about 7 years of hard luck to get this far. So what this is E. coli, which isn't generally considered a bacterium which differentiates. This bacterium is growing in as close to steady state as we could manage it. So all of the parameters, the values of the parameters are kept constant. We've grown them for 15 generations at a very low optical density. We've try to perturb them as little as possible. According to traditional microbiology, the Moeller School back in the 1950s, who did fantastic work, the daughter bacteria, the daughter cells should be the same. The average cell represents the population. It's not what we find. You can be reassured because over the time that we've been doing this, other people have been publishing in the leading journals using other techniques that bacteria grown what they consider to be steady state do exhibit enormous phenotypic diversity. So they're publishing E. coli generates phenotypic diversity. But we've done it a different way and we've been slower to publish. Okay, so what we have here is a bacterium which has been labeled by growing it for 2 generations in nitrogen 15. And then we've analyzed the distribution of this isotope and of the corresponding commoner isotope, nitrogen 14, using a NanoSIMS 50, which is secondary ion mass spectrometry, and essentially involves preparing cells as for electron microscopy, taking sections, and then putting in a high vacuum, and then you have a very high energy beam, in this case of cesium ions, which scans it. There's a sort of collision cascade. It pulverizes the macromolecules and molecules. You have recombination of these molecules on the surface, and then the recombinant ions are analyzed using mass spectrometry. It's incredibly sensitive, so only a few ions can be, you can count only a few ions, down to a few ions. It's actually quantitative over a considerable range. And then you can get the ratio of nitrogen 15 to nitrogen 14, total nitrogen, and you can display it on an image because the technique is relatively precise. 50 nanometers is the name given to it. It depends on the abundance of the isotope. I'm sorry about that long explanation. It's a horrid technique, and it's a machine which costs a lot and consumes a lot of money. So it's one of these, like one of these Greek monsters that feed on their children all the time. Anyway, what we've done here is something very, very strange. We've taken the isotope ratios, we found the median value, and we've divided them into 2 according to whether the pixel is above that value or below that value. Okay, so what we're looking at is the incorporation of nitrogen 15 in different parts of the bacterium and just comparing, is it above or below the median? And we have several hundred, maybe about 500, 600 pixels here. And as you can see, those above the median are this side, this half of the bacterium, and those on the other side are below it, which is very strange and statistically impossible just on a random distribution basis without some sort of dreadful artifact underlying it. One thing that this may represent then is that even in a non-differentiating bacterium such as E. coli, it's getting ready for 2 regimes of growth. The bacterium is generating coherent diversity of growth rates. And we observe a very wide slew of growth rates. This, by the way, this sort of thing is very important because when you treat people with antibiotics, you end up with persisters which are not growing and which can't be affected by the antibiotic. They're not mutants, and when the antibiotic grows, they can start to grow again and repopulate. That's quite a rare event, but what we see is a wide distribution of growth rates. Okay, and it's not that heretical. Okay, so are water structures involved? And I'm going to slowly, 5 minutes. These are very long minutes, I hope. Okay, well, water's certainly different in spores. Okay, so I'll flash over. That's a recent paper. It's a Cell paper. You can see the water inside the spore. Okay, so that suggests that water structures can be different in different cells and in different parts of cells. Okay. Well, I'd like to try and bring this back to hyperstructures. So hyperstructures, big structures which are responsible for different functions. Daughter cells are going to have different phenotypes. They're going to need different sets of hyperstructures. So we're going to have segregation of different sets of hyperstructures with different regimes of incorporation of nitrogen-15, different metabolic rates at different times of the cell cycle. And that's just a depiction to show where maybe different sorts of water structure could help in this problem of producing daughter cells with coherent phenotypes. So you could imagine that one of the daughter cells is going to be going towards growth, towards that basin of attraction, with essentially non-equilibrium hyperstructures coupled transcription, translation, insertion into the membrane. The DNA is in one sort of state versus equilibrium hyperstructures which don't require a flow of energy, and the limit, some of the DNA could approach a cholesteric phase, it's not being expressed. You could imagine that there are different sorts of reactions being favored because of the different preferences for water structures on one side or the other. It opens the realm to coupled oscillations characterizing the different parts. So, of course, water in its different forms, of which I'm hearing about at this conference, could be playing a central role in the organization of these two budding phenotypes. Okay. So, right, in the last 30 seconds, I'm going to explain how the universe began, the nature of God, and where you fit into it. Okay. So the other end of the cell cycle is initiation of DNA replication. How does it actually work? And if you go along to the meetings of the specialists, you can get the impression that we really understand how it works. There's an enormous, very precious quantity of molecular detail. But what exactly is the fundamental signal? And that's not quite so easy to understand. And anyway, why bother replicating your DNA? Let's suppose I'm going to be a bit lazy as a cell, and I'm going to say, I don't believe in all of this crap. I'm not going to replicate my DNA. Too bad. I'm going to be all right. I don't believe it. I'm an atheist. I'm just not going to believe it. Well, what will happen, actually, is that if you don't replicate your DNA, it will become limiting for growth. The RNA polymerases will pile up on your genes, and eventually you won't be able to increase the amount or the rate of production of messenger RNA. Similarly for the ribosomes translating it, you will plateau at a certain linear growth rate. So you do need to replicate your DNA if you want to maintain exponential growth. Okay. So an intelligence system, and I think this applies, so this is a scale-free concept, this applies to a lot of different organizations, is to sense how hard you're working and to change, to make, for example, a new organization, to set up a new shop, to make a new cell at a critical level of intensity of activity of your constituents. The most obvious place to look for that is something called, in the eukaryotic cell, the nucleolus, which is where ribosomes themselves, or at least the RNA which leads to the ribosomes, is produced and where the ribosomes are assembled. These are one of the most important bits of machinery in the cell. They're protein-making machinery, because the cell is mainly made up of proteins. So the nucleolus, and that's a hyperstructure, we would term it a hyperstructure. And the bacterial equivalent, this is the bacterial equivalent, these yellow spots here. This is fast-growing E. coli, and that's where we think ribosome synthesis is mainly occurring, so a nucleolus-like structure. You would listen Yeah, 1 minute. Oh, well, thank you. That was, yeah, okay. I'll speak quicker and quicker. So there is a kind of a nucleolus in bacteria. So ribosomes working. Okay. How could that signal to the initiation or to initiate DNA replication? Okay. How could chromosomes provide what we call it intensity sensing? And this is a wild speculation. Okay. So could the ribosome is going through a fixed simple cycle. The messenger RNA is moving through it to be translated into protein. It's going from the A site to the P site and to the E site. Okay. So that, now, think of Huygens clocks. The clock itself might be complicated, but we've got the pendulum moving and being coupled by the wall. Well, okay, ribosomes are very close together. They are cheek by jowl on the messenger RNA. There are a lot of them. And in principle, they might be able to generate coupled oscillations. So that's what I would like to think, that ribosomes might generate coupled oscillations, and that changes in water might couple the movements of one ribosome to another. I don't know whether it's theoretically possible or not. There's a curious paper in Science a few years ago from the Pelling group where they stuck an atomic force microscopy probe on a cell and they heard it going boom, 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 boom. But I don't know whether that's, well, there isn't much else come out about that. And a wonderful piece of scientific exploration here from Michio Matsuhashi back in the 1990s, who found evidence for sonic communication between bacteria. And actually, Michio did quite a lot of work subsequent to that. I don't know any reason really why that shouldn't be true. I've had long correspondence with him and with people who've known him. It's a very intriguing possibility, and I take it quite seriously. Okay, and I can answer questions about it because I'm going on for too long. So there we are. Our ribosome movements coupled by water. So the take-home message is real. Well, I'll leave the names of the people who did the Sims work. Take-home message is finding coherence is really important for cells in the generation of phenotypic diversity, and maybe water could be involved in it, but it's for you to tell me and not the reverse. Thanks very much, and sorry to have talked over your coffee break, but you didn't really want coffee anyway. Okay.
All right, a couple of questions.
Okay.
Yes.
Norris, that's an excellent lecture. Hello?
Hello?
And it's consistent with your concept of hyperstructure playing essential role in cell biology.
Yeah.
And I think all we need, it seems to me, that your hyperstructure concept is based on proteins, systems of amino acids and so on. But in addition, like all structures in the cell, there may be hydration shell surrounding any hyperstructure in the cell. And this hydration shell is well known in inorganic chemistry to determine rate of electron transfer reactions. So it may well be that the coupling of ribosomes is indeed mediated by water forming hydration shells. So I think all you need is to add to your hyperstructure concept the hydration shell concept, and then I think you can explain a lot of things that you're trying to explain.
That's a wonderful suggestion. I should say that I've concentrated on the protein aspect because that's what Walter Schubert studies with the toponome, which I do urge you all to look at because it may change your view of what cells really are. But in fact, the hyperstructure concept extends to, for example, the coupling between transcription and translation. So messenger RNA, protein, ribosomes, RNA polymerase, the gene at the origin of it, a structured lipids in the membrane. So it's many different types of constituents can constitute a hyperstructure. I apologize if the concept is somewhat vague. I mean, the cytoskeleton, in a sense, in bacteria, the EFTU, elongation factor TU cytoskeleton, there's an actin-like cytoskeleton. We would put those in the domains of hyperstructures simply because, following the advice of my more intelligent and experienced colleagues, it's not a very good idea to be too rigid in your classification of things in the early stages of development of the concept. So we like to have a benefit from this sort of, apparently the term is the generous Darwinian fog. Sorry, okay, another question. Yeah.
Any others? Yeah, here.
In relation to the complexity of a cell,
Yeah.
I think we must treat the cell as a crowded system in which there are contacts with its
I can hardly hear. Sorry, can you say that again?
I think in terms of defining a cell as a complex system,
Yeah.
we should talk about crowded systems in which we have hyperstructure touching each other, and or at least near each other. And in between them, we can find different states of water. One of them is water of hydration,
Yeah.
but the other one is confined water that is thermodynamically labeled to response. And in that sense, the processes in the membrane is very important in order to how the cell, in this case the bacteria, can take the signals from the environment. Your talk is very nice because it's a nice introduction of what I am going to talk after the coffee break. Thank you very much.
Yeah, no, thank you, Hannibal, for those good points. You're absolutely right. There's very little, as I understand it, free water in cells. I mean, of course, we don't look at the ions within a cell. While I'm talking about ions, I did look in Jerry's book recently, and this, when he was giving his talk about like attracts like. As I understand it, ion condensation, as developed in polyelectrolyte theory by Manning and Usawa, allows for ions to exist in three states: bound, free, and condensed. And in the condensed form, for example, in a linear charged polymer, the ions at a certain density of charge, certain value of the parameter of charge, the ions will diffuse along it, and charged polymers with identical signs will come together because they share counterions. And that's the sort of thing that might be somewhat heretical to import into biology. But if it exists outside biology in the world of physical chemistry, I think cells would be foolish to ignore organizing principles that they could use. And as regards your point about the membrane, maybe I'm taking too long to answer, there are all sorts of many types of experiments showing that perturbations of the membrane, its composition and distribution of lipids, have major effects, for example, on the positioning of the division site and the structure that they get spiral structures in a mutant which is rich in polyethanolamine and deprived in cardiolipin and so on. So there's a super marriage to be made between water, lipid structures, and cellular events. Sorry, yeah.
No, let's go. Wait, one more, Nigel?
Just to say that, in fact, by coincidence, I published a paper in 2009 explaining that water is incredibly important for understanding FTSZ. I've emailed you a copy, and in the divisome, I've emailed you a copy, and I'll put a copy up on my webpage for other people to look at. So the answer is yes, it's very important.
Oh, thank you, Nigel. Well, okay, well, we'll talk about it. Fortunately, you've only sent it me now, not back in 2009, I hope, because, you know, I'm not very good at handling my emails, but, yeah, I look forward to reading it. I see that some others of you have got questions. I'd be more than happy to talk with you, perhaps during the coffee break. Yeah. Okay, well, thanks very much for your attention.